First of all, let me thank you very much for your time and your availability. I know that it's 8 o'clock in Hong Kong. It's
Singapore.
It's in Singapore. It's supper time, but I mean we're going to be quick.
Okay.
Luca, you're the best. The mic is yours.
Okay, thank you very much, Prof. So for me, it's a great pleasure to introduce you Dr. Valerie Chew, that is currently a principal investigator in Translational Immunology Institute and an assistant professor in DukeNUS Medical School in Singapore. Dr. Chew's research focuses primarily on understanding the complexity and diversity of the immune context of the tumor microenvironment and its influence on clinical outcome or response to therapy in patients with hepatocellular carcinoma. Their current work involves high and multidimensional immunophenotyping and immunomonitoring of HCC microenvironment, which allows holistic profiling of immune. I was just telling it was a great honor, and because we met during our ILCA International Congress this year and last year, so for me it's a great honor, and so the podium is for you, Valerie.
Thank you. Thank you so much. Thank you so much for the kind invitation and kind introduction. So let me begin by sharing my slide. And I know now it's around maybe 2 p.m. at your time. It's after lunch. So I hope you don't fall asleep. So first of all, my name is Valerie Chew. So with a very kind introduction from Luca. So I'm from actually Translational Immunology Institute, TII, SingHealth-DukeNUS, Singapore. So it's 8 p.m. now, my time. And today I'm going to talk to you about tumor immune microenvironment. So this is my research area. And I'm going to tell you about, understanding of tumor immune microenvironment and how we can use that for biomarkers or therapeutic discovery. So I have no disclosure to start. So I think with a quick introduction to my research interests. So as you all know, studying cancer, there are maybe two general approaches. A lot of people actually focus on the tumor cell itself, which what we call the intrinsic factors. You study the different mutation and different pathway that the cancer cell takes to proliferate or to progress. However, these have not lead to a cure to most of the solid tumors that we know, for example, in HCC. Our team are particularly interested in the tumor microenvironment, or what we call the extrinsic factor. And in particular, my lab is interested in studying the immune microenvironment, which are the immune cells that infiltrate to the tumor microenvironment. And we are also interested in studying the crosstalk between the immune cell and the tumor cell, for instance. So this is my research area, and I focus a lot on liver cancer. So this is the topic of today. So it's time to talk about time, because time stands for tumor immune microenvironment, and this is very important. So first of all, I will break my lecture today to two parts. First, we will talk about understanding time in HCC. Secondly, we will talk about biomarkers and therapeutic discovery with this understanding of tumor microenvironment in HCC. So I think this audience need no introduction into the disease itself. I think most of us probably study HCC one way or the other. You know that this remains as one of the deadliest cancer types in the world, and particularly actually affecting Asia. And you also know that this is the disease with high mortality rate. So the incidence and the mortality rate still almost equal to 1, which means a patient diagnosed with HCC will eventually die from the disease itself. And it remains one of the most common cancer deaths, particularly in males. And with the poor prognosis, as mentioned, even though early HCC, the first line of therapy is usually resection, but we know that a lot of patients actually relapse. And when they relapse, they are presented with advanced disease. And we know also there's limited treatment options for advanced HCC. Not until actually immunotherapy came into the picture. So previously, we only have one drug, sorafenib, which is a TKI for many years. But more recently, we now have immunotherapy as well. So for example, you all know the Iron Grave 150 trial. And now we have anti-PD-L1 with anti-VEGF combination immunotherapy in the clinics, which show very promising clinical outcome. So we actually began our interest in studying the immune microenvironment or HCC long time ago. In fact, in 2010, when I was still a postdoc. So actually, we began studying immune microenvironment in the patient from Singapore cohort. So this is the HCC patient from Singapore. Altogether, we only have like 61 patients. And we found that, however, from these 61 patients, we can see that a number of pro-inflammatory immune genes are important for their long-term survival in these patients who undergone resection. So these are resected tumor and the genes expression in them. So we hypothesized that if we have certain pro-inflammatory microenvironment in this patient, that can actually lead to a long-term survival in this patient even after resection. And I think more particularly with this interest, we then carry on to extend to another 2 external cohorts. So we collaborate with our collaborators from Hong Kong as well as in Europe, at the time Zurich. And then we are able to validate that, in fact, these pro-inflammatory genes, in fact, 14 immune genes, they can actually accurately predict for patient survival in our training cohort, of course, and also in the validation cohort from Hong Kong and Zurich. And so one particular genes or molecule that I want to talk about and emphasize is toll-like receptor 3. In fact, we found that these innate immune molecules have important function in HCC. So through a series of in vitro and in vivo studies, we found that toll-like receptor 3, by activating them, we can actually have different function to induce anti-tumor activities. So first of all, we are able to induce the tumor cell death if we activate toll-like receptor 3. And we are also able to activate the NK cells, so they express high level of toll-like receptor 3, and they can get activated, and they can actually go on and have cytotoxic effect on the cancer cell. At the same time, we also found that toll-like receptor 3 activation can actually induce chemokines secretion, and these chemokines are able to recruit more T and NK cell into the tumor microenvironment, therefore providing more anti-tumor activities there. But I really want to talk about more of our recent work in HCC tumor microenvironment. So ever since I became independent in 2015, I actually joined the lab in TII, SingHealth-DukeNUS, and we use this technology called CyTOF. Maybe some of you have heard of this technology. This is a time-of-flight mass cytometry, and basic information about this is like a more high-dimensional flow cytometry, but we, instead of using fluorochrome tag antibodies, we use the heavy metals tag antibodies to study the different immune profiles in our cells. So we can actually study up to 41 markers, immune markers, intratumoral and surface markers simultaneously. Of course, this is at single cell level. And also we optimize our process to allow us to study low cell number. As you all know that for a small piece of tumor, liver tumor tissue from patient, sometimes we only get quite a few number of cells. However, we have already optimized our process to be able to study even out to down to maybe 100 to 200 cells number. And we also barcode our samples, so we can actually combine a few samples simultaneously to eliminate some of the batch effect. And we use our in-house analysis pipeline. This is actually published in Nature Biotech in 2020 to unbiasedly study the different immune landscape of samples from HCC. So a basic overview of the studies that is carried out in my lab. So we are usually also interested not only in the tumor, we are interested also in the adjacent non-tumor liver tissues. And we also study the blood as a systemic immune system. And we, as already mentioned, subject them to CyTOF analysis. We also use a number of tissue profiling methods, such as the multiplex immunofluorescence using Vectra system. And recently, we also venture into spatial transcriptomics. We also make use of bulk RNA sequencing or single-cell RNA sequencing to understand better the different immune cell or non-immune cell in the microenvironment. With collaboration with Genome Institute of Singapore, we also, as certain study, look into the mutation of the tumor by looking into whole genome sequencing. This is the DNA sequencing. Together, we aim to provide a more comprehensive immune landscape. So one of the earliest study that we published in PMNS is actually we describe the immune gradients across different microenvironments in HCC. So we started off with the PBMC, which is a systemic system. If we compare to the liver or the tumor microenvironment, we see they are more enriched with the naive type of immune subsets. If we move on to the non-tumor liver tissue, of course, we see more memory subtypes or immune subset. But however, we found that they are not really exhausted in nature. But in the tumor microenvironment, indeed, we find that PD-1 expression is increased in a number of immune subsets that we found enriched in the microenvironment. That includes actually Treg cell, effector memory CD8 T cell, and resident memory CD8 T cell. In fact, not only PD-1, we found a number of checkpoint molecules are being highly expressed by the effector memory CD8 T cell and the resident memory CD8 T cell in the tumor microenvironment. And with this preferential expression of checkpoint molecules, we at that time hypothesized that HCC could be a candidate cancer to be treated with checkpoint inhibitor. And of course, later on, you know that in a number of clinical trials, anti-PD-1 does provide clinical benefits for patients with HCC. And also, I would like to also mention, because we found the non-resident and resident memory CD8 T cell are important and enriched in the tumor microenvironment. And this kind of validated by another study later on in Nature Cancer, where they found that both the resident and circulating memory CD8 T cell are important in melanoma patients who show durable response to immunotherapy. Therefore, I think this is probably the same scenario also for the HCC tumor. So moving on from studying the tumor microenvironments, the next question that we ask is, how about different etiologies that can drive HCC? So first study, we actually look into the viral versus non-viral related HCC. So in my part of the world in Asia, we have more patients who are down with HPV related HCC. Therefore, we compare HPV related versus the non-viral related HCC. As you can already see from our CyTOF study, the microenvironment of these two subtypes of cancer are quite distinct in nature. So for instance, if you can see the HPV related HCC, they are enriched with a number of immune cells, particularly the Treg cell, as well as the resident memory CD8 T cell that express higher level of PD-1 as compared to the non-viral related HCC. So for instance, at that time, we already found that PD-1 is particularly enriched in HPV-related HCC, and we predict that anti-PD-1 should work better in HPV-related HCC. Why? Because you already know that anti-PD-1 can reactivate CD8 T cell that express PD-1 in the tumor microenvironment. And in our own study in gut, we also found that anti-PD-1 can reduce the immunosuppressive function or Treg cell in the tumor microenvironment, particularly in the HPV-related HCC. Of course, later on, you are familiar with this paper published in Nature 2021, where they really show that indeed the viral-related HCC does respond better to a number of checkpoint inhibitor in a number of trials. Particularly HPV-related HCC, they are the ones that really respond much better to a number of checkpoint inhibitors, thereby validating our findings. But I would just like to point to you that in this study, our cohort of non-viral related HCC are mainly non-NASH in nature. Therefore, this leads us to a new study right now, currently unpublished. We are wrapping up the story. We look into steatosis-driven or NASH-related HCC. Same thing, we use CyTOF, wild RNA sequencing, single-cell RNA sequencing, and more recently, we also venture into spatial transcriptomics. And we define our patients who are steatosis-driven as having fats more than 5% in the hepatocyte. And obviously, we found some distinct difference in the immune landscape of steatotic versus non-steatotic HCC. Particularly, we found more immunosuppressive subset within the steatotic-related HCC. We also have some spatial data to show this phenomenon. But of course, we are still wrapping up this story, so I will not go into too much details regarding this. So with the study into different etiologies related HCC, we also published our opinion in 2020, and particularly focusing on immunotherapy options for HCC that is derived from different etiology. I think I will just talk more about HPV-related HCC because this is what our study shown. And I would also like to highlight that Treg is particularly increased in this particular subtype and the PD-1 expressing Treg. Therefore, we think that by anti-PD-1 combination, this could work the best for the HPV-related HCC. And moving on from the different etiologies, we also know that another phenomenon can drive immunosuppression in cancer, also including in HCC. This is hypoxia. So as you know, hypoxia is a phenomenon of lack of oxygen when the tumor grows too big, or lack of vessel growth in the tumor. And we look into the different subset of tumors because we know that intratumorally, HCC are very heterogeneous in nature. Therefore, we use actually the gene signature for hypoxia to divide the different regions of the HCC tumor into hypoxia high or hypoxia low. And then we look into their microenvironment and the immune landscape of these particular tumor sectors. As we can find that they are distinct in terms of the lymphoid landscape as well as the myeloid landscape. Particularly, we saw that the exhausted CD8 T cell and the Treg cell are particularly enriched in the hypoxic-related HCC. In the myeloid compartment, we saw that CD169 positive macrophages, which is more M2 kind of macrophages, and HLA-DR low CDC2. And we know that CDC2 actually have the function of presenting tumor antigen. However, in this case, they express low HLA-DR. HLA-DR is actually the molecule for tumor presentation. However, the low HLA-DR means that this CDC2 have already lost the function to present tumor antigen. Therefore, overall, we see a more immunosuppressive microenvironment in HCC that is hypoxic in nature. We also found that this Treg cell actually have a very close correlation with the CDC2 subset, particularly those that have low HLA-DR. And are they really correlate or interacting with each other? Therefore, we actually look into our CyTOF data again. And interestingly, we found doublets, which means there are two cells very closely linked to one another or interacting with one another. And we found that these cells are actually CDC2 with T cell interacting. These are the doublets. And the T cell that is interacting with CDC2 are actually CD4, not CD8. And when we get down further, we found that they are actually Treg cells. Therefore, we found that the Treg cells are actually interacting with the CDC2 subset. And seeing is believing. We also look into the tissue. We stain these two subtypes of immune cell, Treg and CDC2. And indeed, we do find them to be close by to one another, particularly in the region where it's high hypoxic region. And if we measure the distance, as you can see, hypoxia high region, the distance between Treg and CDC2 are much shorter than in low hypoxic region, indicating they are interacting with one another. So of course, the next question would be, what are they doing if they are interacting with one another? In fact, interestingly, we found that they actually undergo this mechanism called trochocytosis. This is a Greek word. And actually, the meaning of it is to eat or nibble off something from the plasma membrane from one cell and transfer to the other cell. And so we found this is really happening between CDC2 and the Treg subset by the in vitro experiment of co-culturing these two subsets of cells together under the hypoxic environment. So we saw that in hypoxic environment, CDC2 subset actually lose the HLA-DR molecule. And this is only occur in the presence of Treg cell. So you can see with the presence of Treg, the CDC2 subset that lose the HLA-DR are more than those without the Treg subset. Therefore, showing that Treg is important in this CDC2 subset cell losing the HLA-DR. And where do the HLA-DR go? In fact, we found that it is actually enriched in the Treg subset. As you can see in the hypoxic microenvironment, the Treg subset actually gained the HLA-DR molecule from the CDC2. So in conclusion, we found that CDC2 actually lost the HLA-DR molecule, thereby losing their ability to present tumor antigen. At the same time, Treg gained HLA-DR, and from literature, we know that HLA-DR positive Treg are also more immunosuppressive, and thereby showing that in hypoxic microenvironment in HCC, this is the mechanism, one of the important mechanisms for immunosuppression in the microenvironment. So as I already showed you our deep understanding into the tumor microenvironment or HCC, we also know that the tumor microenvironment undergo a sort of immune evolution to become more exhausted and immunosuppressive in nature. And I have shown you that multiple factors can drive this immune evolution, including hypoxia, HPV, or fatty liver for that instance. But this constant fight between tumor and immune subset it actually is the main factor that kind of drive this kind of evolution. This is at least what we believe. I think moving on from here, we want to really use this knowledge to also understand the mechanism and the potential biomarkers for response to cancer therapy. So this brings me to the second part of my lecture, and I'm going to talk about biomarkers and therapeutic discovery using our knowledge in tumor immune microenvironment in HCC. So one of the first study by our group is looking into selective internal radiation therapy or SIRT in HCC. This is in collaboration with Professor Pierre Chow, who is a clinical the PI that runs the clinical trial using Y90 radiotherapy in HCC patients. So Y90 radioembolization or SIRT is given as a single dose introduced locally through the hepatic artery directly into the HCC tumor. Since this is only given a single dose and it has a very short half-life of around 3 days, and however, we are puzzled to see the response from the patient. Usually we see a delayed response around 3 to 6 months. And when the patient responds, they often show a sustained response of beyond 6 months. And to an immunologist, I thought this must have some immunological impact or response that we can actually visualize in the immune system of the patient. Therefore, we started collecting the blood before and after Y90 radiotherapy. At the same time, we also collect the tissue from the patient who are downstaged by the Y90 radiotherapy and went for resection after. Again, we use CyTOF and RNA sequencing to study the immune changes. First of all, if we look at the local tumor microenvironment, comparing the Y90 treated versus the treatment-naive control, we can see the immune landscape are very distinct in nature. So for instance, the Y90 treated patient, this is post-treatment, you can see the enrichment on number of cytotoxic and killer types of immune cell. These include the natural killer cell, natural killer T cell, CD8 and CD4 T cell that express activation marker, including the granzyme B and CD69. And in the treatment-naive control patient, we see the usual enrichment of immunosuppressive subset, including the Treg cell, thereby showing that Y90 radiotherapy can actually reactivate the immune system in the tumor microenvironment of the HCC who are responding to the treatment. And what about in the blood? In fact, we look into the peripheral blood. We do see that responder versus non-responder are presented with different immune profile as well. Particularly in the responding patient, we see this so-called pre-activated T cell, and this is what we call the T cell that express PD-1 and TIM3. They are there in the blood even before the treatment is given, and we can still see them out to 3 months after treatment. as compared to the non-responder. So this is very clear difference. And in fact, at the time we thought it would be good to combine Y90 with anti-PD-1 because they clearly express more in the responder. And in fact, this initiated a study in Singapore combining Y90 radioembolization with nivolumab, which is anti-PD-1. I do this in collaboration with a number of medical oncologists from National Cancer Center, Singapore. And I think more importantly, we found this particular T cell express certain chemokine receptor, showing that they actually have a tissue homing capability. So this responding patient in the T cell that express CXCR6 and CCR5 as compared to the non-responding patient. Again, this T cell is already there pre-treatment and as 3 months after treatment. With that, we hypothesize they can home to the tumor microenvironment. Do we have the evidence? In fact, when we look into the tumor microenvironment and compare the Y90 treated tumor versus the control tumor, we see outregulation of a number of immune pathways in the Y90 treated HCC tumor. Particularly is the chemostasis activity. And if we look into the particular chemokines that highly expressed or enriched in the Y90 treated and responding HCC, we found CXCR16 and CCR5. This is actually the chemokines that bind to the CXCR6 and CCR5 on the CD8 T cell that we found in the peripheral blood. Therefore, this actually brings to us to say that these CD8 T cell in the peripheral blood are able to home to the tumor microenvironment through the upregulation of these chemokines that binds to their receptor, giving rise to better clinical response after Y90. We also make use of our SingHealth data before the therapy in order to derive a system that can predict a certain patient will respond or not respond to Y90 therapy. So of course, we use all our single cell data from SingHealth and we feed into the random forest algorithm and derive a particular prediction model to accurately predict which patient will respond or not to Y90. In fact, we found our prediction model is much more superior than any clinical parameters that can be used to predict response to Y90. Following that, we also published a review article because apparently we found that radiotherapy combining to checkpoint inhibitor would probably give an enhanced immune response, particularly in the patient who respond to Y90. And of course, checkpoint inhibitors is so important in the treatment of advanced HCC nowadays. So of course, we also look into the response biomarkers for checkpoint inhibitor blockade in HCC patient. In this case, we look at anti-PD-1 monotherapy, and this is the CheckMate 40 trials. As you already know that we do see around 20% response rate in the patient treated with anti-PD-1 monotherapy. At the same time, we also often observe grade 3 to 4, which is more severe immune-related adverse effect, which is a toxicity experienced by this patient, also around 20%. We also know that the adverse effect actually experienced by patients treated with checkpoint inhibitor are more autoimmune-like. Therefore, we also believe we can actually capture or see this in the peripheral blood of the patient. We again collect the blood from the patient before and after anti-PD-1 treatment. At the same time, we also look at a number of tissue samples that we can obtain from this patient after anti-PD-1 treatment. And we are interested in 2 clinical outcomes. One is response, and one is immune-related adverse effect. And we studied on using SingHealth and single-cell RNA-seq mainly. And the main focus of this study is to maybe if we can design another therapeutic options to increase the response without increasing the adverse effect in this HCC patient. So first of all, when we look into our SingHealth data, this is from the peripheral blood. We already can observe that the immune landscape of responder versus the non-responder are quite distinct in nature. Likewise, we also observe in adverse effect, the patient who experienced toxicity, they also presented with quite distinct immune landscape in the peripheral blood. And I will bring your particular attention because there are so many of them, but we focus in the end at the CXCR3 positive CD8 T cell or effector memory CD8 T cell and antigen presenting cell, because these two cells are actually common immune subset that appears in the response and the toxicity of this patient. And in order to decipher them apart, particularly the antigen presenting cell, they appear in both response and adverse effect. And we use single cell RNA-seq to further decipher their sub phenotype. And in fact, we found that CDC1 is actually related to response, whereas the CD14 myeloid subset are actually linked to adverse effect. With that sense, we can actually uncouple a different antigen presenting cell that is related to response versus toxicity. However, can we find a better treatment for this patient? We then focus on CXCR3 positive CD8 T cell that's effector memory in nature. And this particular subset actually appear in both the response and toxicity. But we hypothesize their signaling pathway must be different between response and toxicity. And true enough, when we look at our cell-cell interaction data, we found that particular pathway is only related to response. This is the TNF and TMF receptor 2 pathway. The TNF and TMF receptor 1 pathway, on the other hand, are related to toxicity. Therefore, we test the different receptor in the mouse system, and this is the mouse model with the HCC or hepatoma tumor. We treat them with TNF receptor 1 or TNF receptor 2 together with anti-PD-1. And look what happened. So we found that anti-PD-1 in combination of TNF receptor 2, this is the one that is related to response. We observed that all the animals do not have any tumor at all at harvest. So this actually gives the best tumor control among different combinations. Even though TMF receptor 1 also shows some control of tumor burden, however, because this particular molecule is linked to toxicity, we actually saw enhanced toxicity, increased liver size and the weight, as well as infiltration of CD8 T cell, particularly activated CD8 T cell in the liver microenvironment, showing that if we block TNF receptor 1, we actually see more toxicity. Thereby, the conclusion from this study is that combining anti-PD-1 and TNF receptor 2 is the best combination option for us to have enhanced response without the increase of toxicity. So this is the summary of the study already published last year. And in fact, when I look into the literature, I found that TNF receptor 2 has been shown to play many different functions. In fact, people have shown that TNF receptor 2 can actually drive tumor progression and metastasis. And therefore, by blocking this, may also have an effect on the tumor cell itself. And it also has an effect on Treg. This is our favorite immune cell. We know that they are enriched in a lot of HCC tumor. And this could also enhance the immune response if we block this particular molecule. And I also look further, and in fact, this particular antibody using anti-TNF receptor 2 is currently being tested in the clinic. In combination with anti-PD-1, there's one study, and this is done in a multiple advanced tumor malignancy, including a few HCC patients, not many, but we are very excited to know this and see whether we can see an enhanced tumor control from combining with anti-TNF receptor 2. I think in my last slide, I just want to talk about the potential of using peripheral biomarkers. As we all know that now the gold standard is still the tissue biomarkers. However, I want to stress that the peripheral biomarkers is non-invasive in nature, it's convenient, and we are also able to study the trajectory or immune changes in the blood. So for instance, in our JHEP study, we trace the different immune subset and we are able to know that they disappear from the tumor, from the peripheral blood. And in fact, these disappearing immune cell actually home to the tumor microenvironment. So when we look at the tumor tissue from the same study, we found that they express certain markers that show us that these immune subsets are home to the tumor microenvironment. For instance, the APC cell, we see a number of antigen-presenting molecules enriched in the tumor microenvironment post immunotherapy. And this is only in the responding patient. We don't see it in the non-responding patient. At the same time, I think more importantly, we see outregulation of a number of chemokines, such as CXCL11, CXCL9, and CXCL10. And these chemokines can bind to CXCR3. And remember, we found that CXCR3 positive effector memory CD8 T cell is also our marker for response that appear in the blood. And this again shows us that this particular subset can home to the tumor microenvironment. And not just us discovering the same similar result. In fact, this study published in Cell recently, they study more than 1,000 patients treated with checkpoint inhibitor across 7 cancer types. And they also found a very strong signature that is related to response. And in fact, there are CD8 genes, and this shows that CD8 T cell is very important. And also, lo and behold, CXCL9, this is a chemokines, as I mentioned, are able to attract the CXCR3 positive effector memory CD8 T cell from the blood to the tumor microenvironment. In HCC, we also see, again, this is Sandro Bruno study in JHEP in 2020, and they also show, again, CD8 T cell is very important. And more recently in Nature Medicine, again, CD8 T cell has been highlighted as a marker of response to checkpoint inhibitor in HCC tumor. So I would like to conclude today's lecture. I have shown you our comprehensive analysis and understanding of tumor microenvironment in HCC. We know that they are very exhausted in nature, and this can be driven by a number of factors, including hypoxia, and as well as chronic hepatitis B infection, and also fatty liver. And we also know that we can make use of our knowledge as immune biomarkers for response to therapy that includes in Y90 radiotherapy, and also for anti-PD-1 checkpoint inhibitor. And we also describe the key roles of CXCR3 positive CD8 effector memory T cell, and we propose a combination of anti-PD-1 with anti-TNF receptor 2 could actually enhance the response without the increase in toxicity. And I would like to thank my group, particularly a few people who contributed a lot to this study, JJ, Feng, Aldo, Yunhua, and Osuga. And my local collaborators, particularly Dr. David Tai, Dr. Shuping Chew, Professor Pierre Chow, and Professor Han Chong To. And I also work with a number of overseas collaborators, including Matthias. He was in University of Zurich before, now in Germany. And I also work with a group from Hong Kong U, including Irene Ng, Thomas Yao, and my collaborator from Korea, including Chang Hoon Yu. And these are my funding information. And of course, I thank you very much for your attention for today's lecture. And I will be happy to take any question from you. Thank you.
I hope you can hear me. So thank you. Yes. Thank you, Valerie. It was a very, very nice presentation and also very interesting and innovative. I think very comprehensive. So now I can open for discussion. So if people have any question.
I just want to make a statement. It's Terry Hines. We're in the University of Kentucky, United States. Me and my students are here, and I just want to say that was exceptional talk. Thank you so much. Just the teaching of it was amazing. I just want to that's all I got to say. I don't have anything else. That was really good. Thank you.
Thank you so much.
Well, I mean, I back up what Terry, what just said by Terry, that was an exceptional lecture. I have just one general question. I'm not an expert in this field, but I mean, do you think that the environment, which actually plays a major role in the development of cancer, is the chicken or is the egg? I mean, yeah. I think that we need to understand what comes first, if there is a first, or it's come together. Because according to this, we can really tailor our treatment according to the stage. And I think your data are very promising in this sense. And I want you to tell us, what do you think?
Thank you I think it's an excellent comment. The chicken and the egg, I think we always cannot really tell which one comes first. But of course, the environment is definitely very important. And we often wonder, like evolution, and one drives the other. If the tumor cell behaves certain way, the immune cell will behave certain way as well. And I feel that it must be the tumor or, your system or the liver has predisposition to cancer cell development. So you will first have the cancer cell, and then the immune cell will try to kill the cancer cell. As you all know, in a normal situation, healthy immune system, you are able to see the tumor antigen and you kill the cancer cell. And then of course, the cancer cells will know that, we cannot survive because the immune system will kill us. They will mutate, and then the immune cell will try to kill them again. And this go on chronically. Eventually, the immune system becomes exhausted. So which one actually comes first? I think probably the tumor cell comes first, because you need to first have the predisposition to have the tumor cell. But again, in our daily life, every day we will have some weird cell or like pre-tumor cells. So maybe it's only during the time when our immune system is weak, then the cancer cell can actually establish itself. Then again, it's the chicken and egg. Is it that your immune system is weak, then you have the cancer? Or you first have the cancer, then you have an exhausted immune microenvironment, or so on and so forth? I do not have the answer to this. I think it will be very extremely interesting to study, like from NASH development into a cancer. I think maybe we can get some answer from that. Or chronic viral infection, how it becomes cancer. Maybe we can get some answer from that. But maybe it starts from information in general. That's what I believe. Yeah.
Thank you
it's
very
the
much.
grandmother of everything.
Thank you. I understand.